Hi everyone, welcome to my channel. Today we will learn about immunity and how to perform this question. So we have done many videos of blood physiology. If you like those videos, please do subscribe my channel. Okay, so let's get started. Immunity, what is immunity? Immunity is ability to fight against infection. Infection can be entry of any pathogen, right? So it is of two type, innate and acquired immunity. Innate is whatever we have to defend against pathogen by birth, so that is innate immunity. Okay, other is acquired immunity, what we acquire after birth from environment or from any infection. So we'll do it later. So innate immunity, so it can be asked as short question, what is innate immunity? What is innate immunity basically? So the immunity we are having by birth. Examples are, for example, skin protect from entry of pathogen. Mucosa, we have mucosa in all over tubings. It can be GIT, it can be respiratory mucosa, which cover all the cells and prevent the penetration of microbes there. Those cells are basically connected with tight junctions, okay, which is also important part of innate immunity. Then we have various secretions like saliva. Saliva has lysozyme, which have antimicrobial property. Saliva also have IgA, which is providing local immunity. Immunoglobulins are present throughout. Then we have tears, which have antibacterial function. We have gastric acid. If we eat food, the saliva encounters, which is again antimicrobial. Then gastric acid, which is having less than 2 pH, which kills all the bacteria. This much acidic pH can kill the bacteria ingested. In vagina also pH prevent infection. Then we have inner gut. There are various floras, which are basically good bacteria which prevent growth of bad bacteria in our gut lining. And not only this, but various functions the good bacteria are playing in gut. Then we have epithelial lining everywhere in every system, which is acting as barrier, and cilia are present on epithelial lining, through which various small microbes, they can be thrown outside by reflexes, for example coughing, sneezing, right? Then we have various cells by birth present, neutrophils, which are first line defense cells, basically responsible for acute infection defense, right? Then we have macrophages, second line defense, where and monocyte cells basically move to various sites in our body and become permanent macrophages there, so they live all our life there and play role for immunity. For example, in liver, those macrophages are like Kupffer cells. In lungs, there are alveolar macrophages, the PAM, pulmonary alveolar macrophages. In brain, we have microglia. And then we have intestine, we have antigen presenting cells. And the most important, complement system, which can't leave, will make a separate video for this. Multiple proteins are present in our blood, which get activated in cascade, which help in ultimately stimulating phagocytosis, chemotaxis, and make pore forming and damage the cell, and activate immune system by making antibodies. Basically antigen antibody complex, when combined, it activate complement system for further phagocytosis and kill the pathogen, right? So we'll make a separate video for complement system. So this is all about innate immunity. Now let's see acquired immunity. Acquired immunity is all the immune responses which occur because of environmental influences. So adaptation to all the environmental conditions, basically. It can be cell mediated, it can be humoral. Humoral is when mediated by B lymphocyte. Like B was basically invented from bursa of one animal. So that's why B is named as B lymphocyte. Then it basically transform into plasma cell and mammary B cell. Plasma cell who make antibodies and mammary B cell, which basically prevent for further infections. Then this is cell mediated immunity. So here we'll talk about cell mediated in detail. Humoral and complement will make separate video for that. So cell mediated immunity, which is mediated by T lymphocyte. Okay, T lymphocytes formed in bone marrow, they move. These T lymphocyte move to thymus, so which is basically giving the name to this lymphocyte, T lymphocyte. Okay, then they live in lymphoid organ. Types are T helper cells, T cytotoxic cell, suppressor T cell, and memory T cell. Now let's see how immune response occur. When a bacteria or any pathogen enters into our body, the first line encounter is basically the antigen presenting cell present in your body. It can be dendritic cell, it can be macrophages, or it can be sometimes B lymphocyte. Okay, so let's see now. Then antigen presenting cells, they engulf the antigen and present it to T lymphocyte like this. This is antigen presenting cells. Okay, and they have engulfed this antigen, and now it has basically MHC2 complex. What is MHC2? It's major histocompatibility complex, this one, MHC2 complex. MHC2 complex, on its base there is a HLA antigen present. HLA is what? HLA is human leukocyte antigen. Human leukocyte antigen, which is part of MHC2, it binds to that antigen and help in expressing that antigen on MHC surface of antigen presenting cell. So this is basically human leukocyte antigen, which basically distinguished or recognized whether this antigen is self or non-self. If it's non-self, this will be presented on the surface of MHC2 complex. Then what happens? Exposed to T lymph helper cells. So this antigen presenting cell have MHC2 here and antigen presented by this antigen presenting cell. Now it basically exposed to T helper cell. Now this is a T helper cell having a receptor which is called T cell receptor. Okay, so T helper cell which has a receptor, T cell receptor, which bind to this MHC2 complex. Now it causes T helper cell activation once these two combine and attaches with various adhesion proteins. So these are the adhesion proteins which makes them adhere to each other, and now once this interaction occur, there is T helper cell activation occur. Okay, what that mean? T helper cell activation means it releases various interleukins, right, and which basically stimulate T helper cell, first thing. And then T helper cell, what it does is it stimulate B lymphocyte to become plasma cell and form antibodies, and antibodies basically do further complement activation and phagocytosis. On the other hand, it stimulates cytotoxic T cell which engulfs the antigen and kill the antigen cells. That's why we call them killer T cell. Okay, so this is a brief about how immune response occur. So let's proceed to this. What happens next? So antibodies, they stimulate the complement system with various proteins. They make pore in the bacteria membrane and they coat the antigen for phagocytosis, making them tasty, and then chemotaxis to attract the phagocytes there. Antibodies kill the antigen by antigen antibody reaction, and they form memory B cell for future infection. In future, memory B cell attacks the antigen if exposed. Okay, so this is a brief video about immune responses, and we have seen a brief description of how the cell mediated immunity occur. So in next video we'll see how complement system is activated and how it kill, basically it help in phagocytosis. Thanks for watching guys. If you have any query, you can comment below. Thanks for watching. Happy learning.